Assalamu alaikum students, how are you all? Hope you all are good and doing well. So students, we have started our second chapter, which is life and living things. Our last topic was about introduction and life processes that has been taking place in living things. And our today's topic is about cells. So first of all, we must know that what is cell. Student, cell is the basic structural and functional unit of life. Without cell, a life cannot exist. So, let's read it. All living things are made up of one or more cells. Some living things, such as bacteria, exist as a single cell. These are called unicellular organisms. Most animals and plants, however, are made up of millions of cells joined together. These are called unicellular organisms. So, what are unicellular organisms? Unicellular organisms are basically those that consist of only one cell. They don't have more than one cell, while the others are multicellular organisms. Multicellular organism means they have more than one cell in their body, and every cell is specialized for performing some specific function. Cells are microscopic. What does that mean? It means the cells are too much small that we cannot see it our naked eyes. We always need some microscope to see these tiny objects. So the microscope, whenever we look through the microscope, the objects or the things seems bigger than their actual size. So that's why we also see or observe the cells under the microscope. It basically magnifies what we see. The most common type of microscope is called a light microscope. Basically, the microscope that we use in our labs is basically a light microscope because it uses light to illuminate the object. Illuminate, like the light just passes through the object and we can easily find out or distinguish between each and every cell or part of the object. So in this diagram, a light microscope is shown along with its function. Students, this is an eyepiece or lens through which we can see. This is objective lens. The objective lens is basically those kind of lens which we can adjust by ourselves. If we want to magnify at 10, so we can change the lens from some other degrees to 10. And if we want to see the object under 20 power, we can change its power from 10 to 20. Other than that, there is a stage. Stage is that place. It's the place where we keep our slide or object which we have to observe. And stage clips are the clips through which we adjust our slide. This one is fine and coarse knob, which is also known as focusing knob. Through it, what we do, we just focus on our object by rotating it. We can take the object close or far from us. This one is diaphragm and this one is mirror, through which light can pass easily. Now let's read it. The object is put on a glass microscope slide and usually covered with a cover slip. As I told you that the object is put on glass microscope slide, this is the stage where we keep the slide. The slide is placed on the stage of microscope and held in place with stage clips, and then we adjust them with the help of stage clips. The hole in the stage allows light to pass through from a light source below. This is the hole through which the light can pass through the object easily. The diaphragm can be adjusted to allow more or less light to pass through the hole. It allows the light to it adjusts the light according to the situation. Light shines through the object and into the objective lens. Then it shines through the object and objective lens that we are using. Light passes from objective lens through eyepiece lens and into your eyes. Through this, the light can pass through objective lens and then pass to our eyes. The focus knob adjusts the distance between stage and objective lens to create a clear image. This focus knob can help us to create a clear or focused image. And the objective lenses are of 3 powers, like 4 powers. They may be in the 5 magnification power, 10 magnification power, 20 magnification power, and 40 magnification power. If the eyepiece magnification power is about 10x and the objective power is 20x, so we multiply both these magnification powers and we will magnify the object up to 200 times. Did you know an electron microscope achieves a much higher magnification than a light microscope? This is because it passes a stream of electrons through the object instead of light. Electron microscope is basically more advanced form of microscope. In light microscope, light is passed through which we can clearly see or observe the object, but in electron microscope, a beam of electron is passed. So through this process we can easily see the objects more distinguished and more clearly. So students, you can see in the diagram that there is an animal cell and there is a plant cell. This is a picture that has been taken from the jaw cells of an animal, and this is a picture that is being taken from the leaves of plant. So students, animal and plant cells have their similar functions almost, like they helps us to grow, they help us in taking food, getting rid of waste, and most obviously they help us to reproduce, but their structures are somehow different from each other according to their body. So the cells are which are taken, or this is basically the microscopic pictures of animal and plant cell. Sorry, these are not the jaw cells, these are cheek cells, and these are the cells from the leaf of a plant. There are many things or many structures that are similar in both plant and animal cell. For example, nucleus, cytoplasm, cell membrane, and cell wall. These are basically cell wall, cytoplasm, nucleus, and cell membrane. These are the structures that are commonly present in both animals and human. What is nucleus? Nucleus is basically the control center of the cell. The nucleus contains information which controls everything that goes on in the cell. Everything that happens in the cell, that information is just stored in the nucleus, and this information is passed from one generation to another generation as well. The next one is cytoplasm. Cytoplasm is all the living matter except for the nucleus. It is a jelly-like substance which is a fluid. Cytoplasm is basically a jelly-like substance, a liquid or jelly-like substance. So it can flow, and it is all the living matter. Then cell membrane. Cell membrane is a thin skin around outside of the cell. It prevents the cell contents from escaping, but it does allow substance like water and small molecules to cross in and out of the cell. Cell membrane is basically a membrane which is present around the cells, and it prevents the cell from breaking or collapsing or shrinking and escaping the food. Like, it does not allow any food particles to pass through it, but the thing that it allows to pass through is water and some soluble small molecules. That's it. So these are the common structures that are present in both animal and plant cell. Now let's coming towards those structures that are only present in plant. So these are cell wall, chloroplast, and vacuole. What is cell wall? Cell wall is basically a rigid firm coating around the outside of the cell membrane. It is in plant cell that keep its shape. Cell wall basically maintains the shape and structure of the plant cell. Next one is chloroplast. What is the function of this chloroplast? Chloroplast has basically a chemical inside it which is known as chlorophyll, and this chlorophyll helps the plants to make or synthesize their own food through photosynthesis. Other than this process, it also imparts or also gives the green color that we see in plants. The green color that we see in leaves is just due to this chlorophyll that is present in the plant. And obviously the main source of photosynthesis is chlorophyll. And what is photosynthesis? Photosynthesis is basically the process through which plants make their own food in the presence of sunlight. The next one is vacuole. Vacuole is a large space in the middle of cell. It is filled with a fluid called cell sap. Vacuole is present in center of cell. What is the function of vacuole? It stores the food for plant cells, for plants, and it is in the middle cell, or the thing which it is filled with is known as cell sap. Okay students, let's discuss test yourself number 2 of chapter number 2. Why is the nucleus called control center of the cell? Nucleus, which is control center of the cell. The nucleus contains the information which controls everything that goes on in the cell. Question number 2 is, what is the cytoplasm? The cytoplasm is all the living matter except for the nucleus. Part B of question number 2 is, what is it like? Cytoplasm is a jelly-like substance which is fluid, means which can flow. Question number 3 is, what is a cell vacuole? The vacuole is the large space in middle of cell. It is filled with fluid called cell sap. So students, Allah Hafiz, take care. Thank you so much.